Welcome to the 19th here Exposomics webinar. My name is Dinesh Parupal. I will be moderating this webinar. I'm from Icahn School of Medicine in Mount Sinai, New York. Today's speaker is Dr. Weihsueh Chiu, professor at Texas A&M University. Before we start the webinar and I introduce the speaker, one slide on the logistic that how we will handle the Q&A. So in your online team, there is a Q&A box right here. And when you click on that one, it will open a side window where you can type in question, and we will answer all the questions at the end of the webinar. So with that, I'd like to introduce our today's speaker. Dr. Weihsueh Chiu is a professor in the Department of Veterinary Physiology and Pharmacology at Texas A&M University. It's really a great pleasure to have him present this webinar. Before joining the university, he worked at the U.S. Environmental Protection Agency, EPA, for more than 14 years. He's a renowned expert in human health risk assessment. His research focuses also on toxicokinetics, physiologically based pharmacokinetics modeling, dose response assessment, characterizing uncertainty, variability, cumulative impact. And he has co-authored close to 200 peer-reviewed publications. Dr. Chiu has participated in several review panels and multiple government agencies, including NTP, CAL, EPA, the FDA, ATSDR, and also international organization, WHO, Health Canada, and OECD. And also participated in several panels and led several panels at U.S. National Academy of Science, Engineering, and Medicine. Dr. Chiu received his Bachelor in Physics from Harvard University, MA and PhD in Physics from Princeton, and have a certificate in Science, Technology, and Environmental Policy from Princeton School of Public and International Affairs. It's really an honor and pleasure to have Dr. Chiu present this webinar. Dr. Chiu, floor is yours.
Thank you very much for the introduction, Dinesh, and it's really a pleasure to present this webinar on this illustrious series. Let me get the sharing going. All right, how's that? Everyone see that?
Yes, it works.
Okay, great. So as I often do in my presentations, I'm going to start with some acknowledgements. So first of all, current funding from the NIH, NIEHS, our P30 Center, our Superfund Center training grant, and then R21 emergency response. Also, I'm part of the interdisciplinary faculty of toxicology, and Professor Natalie Johnson is the chair of that, and Professor Ivan Rusin is the PI of our T32 training grant. And then there's a number of past sources of relevant support that have contributed to the ideas that I'll be expressing today, and they're listed here at the bottom. So just the outline of this presentation. First, I'm going to sort of first go back to basics in terms of the source to outcome continuum for chemical stressors, and then the role of risk assessment and risk management in making environmental health decisions. Then I'll talk about how I think that the exposome can improve risk assessment, and then how it can also contribute to going beyond risk assessment to cumulative impacts assessment. I'll talk a little bit about the National Academy's report that we just released a week ago on this the state of the science and future of cumulative impact assessment. And then I'll conclude with sort of a summary of how I think exposome research can make a difference now in terms of informing environmental health decisions. So what is this source to outcome continuum? Well, it begins with an environmental release of a contaminant that gets into the environment. So there's different concentrations in the environment. Then people are exposed to the different media in the environment, whether it's dermally, orally, or through inhalation. That chemicals then go into the body, and there will be some target organ dose that will then cause some earlier biological effects, and then ultimately adverse outcomes. So this is sort of the sort of scientific backbone of how chemical stressors cause effects, and the sort of translational backbone is risk assessment, where it begins with the process of hazard identification. Where does an agent cause a particular adverse effect? That then leads to dose response assessment. What is the quantitative relationship between dose and incidence or severity of effects? That's complemented with exposure assessment, which is how exposures, how that agent is, how many people who's exposed to that particular agent. And then that's combined into a risk characterization. What, given current exposures or anticipated exposures under different conditions in the future, what would be the likely incidence of adverse effects in the population? Now, risk assessment doesn't live by itself. It is informed on both ends from, on the one hand, research, data and models and measurements that inform the different components of risk assessment. And then on the other side, risk management. What are we going to do about it? And this is where the environmental health decision-making occurs. Do we take specific actions to have specific policies or decisions or regulations in order to address the effects of environmental contaminants? So if we put these two things together, we can see that the source to outcome continuum here is that the first parts are about exposure assessment, from external exposures to internal exposures at target tissues and systemically in the body. Hazard identification then is relating what's in the environment or what people are exposed to or what exposure is inside the body to ultimately adverse outcomes. And then dose response is the quantitative characterization of that process. And then risk characterization then, of course, is putting all these things together. And risk management occurs also along this source to outcome continuum. So you can have risk management that at the environmental release stage, banning a chemical like no more lead in gasoline, regulating releases of chemicals or agents into the environment. You can have risk management at the sort of fate and transport stage, where you regulate how things are disposed so that they won't leave the site of, they won't be released and go into other parts of the environment. You can also remediate the environment in terms of contaminated sites, like removing contaminants from the environment. The next stage in risk management could be mitigation, whereas, for instance, drinking water treatment or land use controls to reduce the contact between individuals in the population and those chemical agents. And then you can have risk management at the sort of exposure step, where you're either reducing contact between the environment and individual, for instance, having personal protective equipment, or reducing absorption, like clay-based sorbents that will reduce what's in the environment gets into the body. So then, where does the exposome fit in? And before getting into that, I want to go back and then again back to basics. Well, what is the exposome? So I really like sort of the Chris Wild's definition in 2012. This is sort of the second big exposome paper that he put out, in which he divides the components of the exposome into three components. So there's the general external exposome, things like economic and social determinants of health, natural and built environment, air pollution, to some extent it goes there. There's also the specific external exposome, where individuals are which is a more individual level of exposure in terms of, say, infectious agents, diet, specific chemicals or pollutants that are more highly localized or in someone's microenvironment. And then there's the internal exposome. This could be metabolites or chemicals that get into the body, or metabolites that are produced. Could be clinical biomarkers, measures of inflammation, et cetera. And all of these, of course, are overlapping. They're not totally separated, and they all have, there's an interplay of these in health and disease. So this suggests that in the source to outcome continuum, the exposome is really anywhere along the spectrum from environmental releases to maybe early biological effects, not really including, just not including this adverse outcome. So what did Chris Wild say about how the exposome can, go from concept to utility? Well, first, yeah, this is just some quotes. He says the concept of the exposome was developed to draw attention to the critical need for more complete environmental exposure assessment in epidemiologic studies. So we can get better epidemiologic data using the concept of the exposome. A second quote, I forgot to put the quotation mark, sorry about that, is that this is the opportunity to sort of link not only exposure and disease, but provide insights into the mechanisms by which exposure might be causing these diseases. And he notes that these mechanistic insights can contribute to the weight of evidence for assigning causality in these associations, because of course, observational studies ultimately are studies of association, but mechanistic data can provide confidence that that association is actually causal. And also he talks about opening up avenues of prevention through modulating specific biological pathways. So in terms of mechanisms and epidemiologic studies, then that's really, in terms of relating this back to risk assessment, that's really improving our identification of hazards. So as an example, I'll use the WHO International Agency for Research on Cancer on how they classify agents as a demonstration of how exposome data can be sort of integrated into this sort of hazard identification process. So IARC monographs basically are reviews of the literature on whether an agent causes cancer. And the review process is governed by what's called the preamble to the IARC monographs. And the first one was in 1971. And this preamble has been periodically updated, and the last update was in 2019. Beginning with volume 124 of the IARC monographs that was from 2020. So within this preamble, it defines how evidence of cancer is considered. So there's three different sort of streams of evidence. There's cancer in humans. So this is epidemiologic studies, but studies of the actual tumors, cancer in individuals, not the sort of intermediate events. There's cancer in animals, and this is typically laboratory rodent studies, animal bioassays. And then there's carcinogenic mechanisms. And since about 10 years ago, there's been a move, and this has been since sort of codified in the IARC preamble, to organize these carcinogenic mechanisms into what are called the 10 key characteristics of carcinogens. And this was developed about 10 years ago. Martin Smith led this effort, and the way it was put together was looking at, well, if we know we know what agents are known to be carcinogenic. So what are some common mechanistic characteristics of those agents? So this is definitely distinct from, people talk about the hallmarks of cancer. So that is characteristics of cancer cells and cancer biology. This is the characteristics of agents that cause cancer. So, for instance, they might be electrophilic, and there's undergo metabolic activation, genotoxicity, altering DNA repair or genomic instability, inducing epigenetic alterations or oxidative stress or chronic inflammation. Immunosuppression is another characteristic of certain agents. Receptor-mediated effects, again, another characteristic causing immortalization. For instance, a lot of the viral caused cancers are due to immortalization, and then altering cell proliferation, cell death, and nutrient supply. So nutrient supply would be things like angiogenesis. So this was really an empirical approach looking at what is known to cause cancer in humans and what are these characteristics of those agents. As you can see, these are all either adjectives like is genotoxic, or verbs, induces epigenetic alterations, and it's phrased that way because these are characteristics of those agents. So there's been a number of papers, many series of papers published since about how to use the key characteristics to identify carcinogenic hazards. This is just a sort of a graphical depiction of the 10 key characteristics with some, common agents, depicted in the middle. Now, a certain agent might have more than one of these key characteristics, or it might predominantly be one of these. There's not a checklist to have all 10. It's not, and it also there's a lot of expert judgment involved. Just because it has one doesn't mean it's necessarily a carcinogen, but it really adds to the strength of evidence for mechanistic basis for carcinogenesis caused by the agent. So how does this relate to the exposome? This is a recent monograph that I participated in, volume 135 for PFOA and PFOS, and these are actually two exposome studies that were included in the literature review. The first one on the left is about inflammation, and the one on the right was about other epigenetic drivers. And so these really are part of the evidence base, for instance, for whether PFOA and PFOS induce chronic inflammation. Now, it was concluded that overall the evidence was not informative enough to conclude that it does cause chronic inflammation in exposed humans. But this is certainly a place where these exposome studies can play a role. The second here is about epigenetic alterations. And again, it turned out that for this particular study, this was, there were other studies that had stronger evidence, but this particular exposome study was considered having inconclusive results or limitations in the study design and were less informative as to the induction of epigenetic alterations. So in addition to cancer, there's been progress in developing these type of key characteristics for a whole host of different hazards. So beginning with the Martin Smith paper published in 2016, I think it was available online in 2015. So sometimes you'll see the 2015 there. Cardiovascular, endocrine disruptors, female reproductive toxicants, hepatotoxicants, immunotoxicants, male reproductive toxicants, and then metabolic disorders. And I think they're working on the one for neurotoxicants in the moment. So if you review all of these, you'll notice that there are some, for instance, inflammation that might be part of multiple sort of hazards, and then others like genotoxicity, which is specific to carcinogens. But certainly this provides a framework for organizing the types of mechanistic data that exposome studies can investigate. So in terms of how exposome studies can inform hazard identification, really the need is for strongly designed studies that characterize these relationship between the external and the internal exposome. It could be with or without the additional associations with overt disease. But you can use then these key characteristics to help organize this internal exposome data so that it can be more easily and more directly inform the actual hazard identification process. And one example is this is a paper that with Ivan Rusin and one of his postdocs, as well as Fred Wright, that basically reorganized transcriptomic data into gene sets that corresponded to key characteristics across all the different key characteristics that were published previously. So that demonstrates how even transcriptomic data can be organized along these key characteristics in order to inform hazard identification. Now, I want to know I know people, it's always good to have a little bit of competition. And so I will say that it seems like exposome studies are kind of lagging behind sort of in vitro and in silico approaches for informing this type of mechanistic data evaluation. This transcriptomic data, a lot of it is from rodents or even in vitro studies, and then there's a lot of in silico efforts as well to understand mechanisms of agents. So it's sort of a charge for the exposome community to really better target these sort of mechanistic data in a way that can more directly inform the risk assessment process. So another part of quote that I like from Chris Wild's paper is that he says, in principle, the exposome could be thought of as an accompaniment to the genome to inform personal decisions about lifestyle or medical interventions. And that is sort of illustrated in this Hallmarks of Precision Environmental Health diagram that's from Andrea Baccarelli's paper from 2023. We're looking at elucidating mechanisms, understanding bases of inter-individual differences, developing biomarkers, advanced risk predictions, sort of multi-omic risk scores like polygenic risk scores, but for exposome data. And then centered here is development and delivery of interventions to prevent disease tailored to the individual. Now, and I tend to agree with Chris Wild on this. I think actually, this is a little bit of a misguided approach in terms of the precision aspect. And he even writes, this is 13 years ago, I do not believe this should be the goal of exposome-oriented research. And it's more likely that this information will be useful at the population level and not the individual level. So certainly elucidating mechanisms is important, understanding the basis of inter-individual differences and susceptibility is important, development of biomarkers of exposure and disease risk is important. But I think from these three, we don't necessarily need to get to the risk scores and the development of personalized medicine type of precision medicine type of approaches using the exposome. Rather, you could taking this sort of mechanisms of inter-individual differences. This was a paper led by Lauren Zeiss in 2013 about how essentially these aspects like the exposome, although we didn't actually use the exposome language in this paper, but things beyond heredity to sex, life stage, existing health conditions, co-exposures, nutrition, psychosocial stressors, these all impact how susceptible people may be going through this source to outcome continuum, from exposure to pharmacokinetics, pharmacodynamics, and then how biological responses then manifest into adverse health outcomes. So really, you could that idea of the exposome being a driver of variability and susceptibility is one in which actually can be interfaced with the risk assessment process. Particularly not just in hazard identification, but in dose response assessment. So with that, I want to go back again to review how dose response assessment is typically done in risk assessment and how variability is addressed. So usually you begin with experimental animal data. I know there's an effort to go beyond experimental animal data, but suffice to say, most of the regulatory policies and risk assessments are still based on experimental animal data. And I'll get to some of the limitations of that in a little bit. But you start with experimental animal data, you identify what's called a point of departure. It's better to actually fit a curve to this, but I didn't want to overcomplicate the graph. But let's just say you have a NOAEL, which is a dose or concentration at which there's no observed effect in the experimental animal. Then you need to extrapolate from animals to humans, say, with a factor of 10. And then you need to extrapolate from, the typical individual to a sensitive individual by additional factor of 10, accounting for human variability. And this leads to what's called a reference dose that's intended to be protective for a diverse population against these adverse effects. So this factor of 10, this does seem kind of crude, but it's still the standard practice now, unless we can get better data-informed information to go away from this factor of 10, whether it's human data or experimental data or even in silico approaches. So, again, the idea of this factor is to protect sensitive populations, meaning that because of differences in the response due to difference in toxodynamics and differences in toxokinetics, people will vary in their sensitivity. And so you want to And these differences can arise, and this is, EPA's definition, right? So that from factors including life stage, genetic polymorphisms, as well as disease status, nutritional status, et cetera. So this is essentially, the exposome, everything except whatever chemical that what chemical or agent that you're deriving this reference dose for. So essentially, those sources of variability in terms of the exposome are influencing the toxicokinetics or the toxodynamics of after chemical exposure, leading to differences in susceptibility to toxic effects. So what has been, first, this factor of 10 is typically divided into two factors of 10 to the square root of 10, or 3.16. And, the idea being that across, for instance, for toxicokinetics, for the same exposure, say concentration in air, people have different internal blood or tissue concentrations, and those that are at the higher percentiles here will be more susceptible. Similarly, for toxodynamics, for a given concentration in the body that leads to sort of the toxicity resulting from a particular concentration will differ depending on one's susceptibility. So the same concentration will lead to different levels of severity, or, to flip it on its side, a given level of severity will be caused by different concentrations across the population. And so there are efforts to replace these default factors of 3.16 with sort of data-derived factors. And this has been in guidance from the WHO since about 2005. A lot of the work, however, has been on toxicokinetics rather than toxodynamics. But the general idea is that you want a factor that will protect a large fraction of the population, and you can use data from experimental or observational studies to try to inform how big that factor is. Because it will likely not be the same for different agents, not be the same maybe even in different populations. So there was a recent paper, just last year by Anna Kreutz describing, in terms of population toxicokinetics, the different factors that contribute to toxicokinetic variability. So there's, of course, physiological differences. People have different lip size for their liver, different cardiac output. There's genetic variability, for instance, maybe an enzyme efficiency. So aldehyde dehydrogenase is a classic example. There's ontogenic differences. So depending on life stage, there's going to be differences in gene expression and enzyme expression. And then there's exposomic factors, other things that you eat, your other exposures to other stressors, drugs that you take, consumer products, other exposures, physical activity, all smoking. These are all can affect the toxicokinetics of for the individual, leading to variability across the population. Now, there's much less on the toxodynamic side. I've been fortunate to have collaborated with Yvonne Rusin and others on this topic to try to use in silico and in vitro approaches to better understand toxodynamic variability. And I just want to illustrate one of these in vitro and silico approaches here. This is a study, again, about a decade old, and I'll talk about some more recent data in a second. But taking essentially cells from a genetically diverse population, this is from the 1,000 Genomes Project. These would be turned into immortalized, so they can be, basically reused and tested in vitro. And then this is a cytotoxicity screening assay, where we took 170 different compounds, looked at the concentration response for cytotoxicity for all these agents across these 1,000 individual cell lines. So each of these graphs shows the cytotoxic response across these 1,000 individuals for a different agent. Sort of organic and inorganic mercury compounds tend to have a very wide variation in terms of how variable different cell lines are to the cytotoxic effects. Whereas, here at cadmium chloride, first of all, it has a lower potency, but it also has a lower variability. And, similarly with catch-all here, this is about the default amount of variability of threefold. So we then looked at across these 170 chemicals and compared it to a fairly limited set of 40-some in vivo studies of toxodynamic variability that was collected by Dale Haddis, and we found a very similar distribution across chemicals. So, the typical chemical would have about a factor of 3, which happens to be default. But there is a large tail in which there are chemicals and agents with much larger degrees of variability, as you can see for these mercury compounds, 8 or 10 or more fold variation across the population. So this is just sort of the pioneering approach to try to address toxodynamic variability, and subsequent analyses have looked at induced pluripotent stem cell-derived cardiomyocytes as a sort of phenotype-specific toxodynamic variability. So whereas this was cytotoxicity, these are more functional endpoints. And then we used this in a couple case studies of mycotoxin as well as some Superfund chemicals. This data was based on the same model of the lymphoblastoid cells of, with about 100 different individuals to try to estimate this degree of variability. Now, this obviously has limitations. First of all, this is only genetic component of variability. These cells are treated exactly the same in every other way. There's no social determinants, no co-exposures, and no disease status in any of these cell models. So there's definitely a potential role for exposome to try to expand the degree to which we can characterize variability in this toxodynamics. And then the other limitation is, unfortunately, there hasn't been any example of this type of data used in any type of decision-making policy, regulation, or action. So in terms of needs from exposome studies to better inform variability and susceptibility, we, of course, first need strongly designed studies to identify subpopulations that have greater susceptibility to chemical exposures. This could be genetic, but in terms of exposome, we're really looking at those non-genetic factors that might increase susceptibility. Just as a sidebar, there's an example that at our toxicology seminar a few years ago, that looking at co-exposure of VOCs and high-fat diet for inducing non-alcoholic fatty liver disease, and without the high-fat diet, there was no impact of the VOCs, but the combination actually led to a potentiation of the induction of non-alcoholic fatty liver disease. And so I'm sure there's other examples of these sort of co-exposures that are potentiating effects that you wouldn't see with just the individual chemical exposure. And then the other is really trying to quantify these impacts, not just identifying greater susceptibility qualitatively, but trying to quantify how big of a degree of variation there is in the population, either from toxikinetic or toxodynamic variability. And I just did a very basic PubMed search. Obviously, this is not a systematic review, but exposome and toxikinetic variability, 8 results, exposome and toxodynamic variability, 2 results. So there's definitely room. And again, just to have a little competition, there does seem to be, again, exposome studies seem to be lagging behind these sort of NAM in vitro and silico approaches for addressing variability. So we talked about hazard identification and dose response. What about sort of the most obvious fit for exposome, which is exposure assessment? And so for this, I want to get back to sort of the ultimate chemical risk assessment challenge, which is, a gigantic chemical inventory, 86,862 chemicals as of July, of which 42,578 are active. And a very small number of these have in vivo animal data, even less with human data. And then even a smaller percentage have been reviewed by authoritative bodies as to their potential hazard. Now, a lot of the action in toxicology has been dominated by these sort of new approach methods to fill this gap, whether it's microphysiological systems using curated databases, transcriptomics, QSAR models, high-throughput in vitro testing, use of in vitro and vivo extrapolation. But these are really focused on hazard and dose response, not on exposure. So essentially, we have an exposure gap in that, for all these new approach methods, the best we can get to is these estimates of chemical potency up here, but we need to be able to compare with exposures to determine what the level of risk is. Are the exposures much lower than the potency of the chemical, or are they overlapping? So the best current quantitative hazard models can predict, for 800,000 chemicals that are on the EPA dashboard, sort of a population median oral dose potency to within about an order of magnitude, so tenfold in each direction. And then based on the studies that we've conducted, at least for the genetic component of variability, that uncertainty is, less than 100-fold or so. Now, we could use biomonitoring data, but current biomonitoring-based exposure data is limited just to a few hundreds of chemicals. So that's sort of, 3 order of magnitude too small, compared to the number of chemicals for which, we would like to have potential exposure data. And then the best in silico exposure models are really quite poor, with a population median prediction of, 100-fold in each direction, or a two-tailed confidence interval of 10,000-fold or more. And then not to say anything about population variability, can't even, predict anything about population variability in exposure. So this is really an opportunity for the exposome research to try to fill this sort of exposure gap in our understanding of chemical risks. So just as a side example from work at Texas A&M, this plastics recycling fire in Richmond, Indiana, about a little over 2 years ago, we have a PTR mounted on a van that went to sample and used both targeted and non-targeted analyses to show that there's elevated VOCs within and bordering the evacuation zone. And particularly for the non-targeted effects, well, what do we make of this? Well, the best sort of comprehensive database is, again, the EPA chemicals dashboard. And about 50% of the putatively identified VOCs are lacking even data on acute toxicity, skin, eye, irritation, inhalation. And this is obviously a concern for emergency response. And then the other limit, on the other hand, the limitation, so this is a limitation in hazard data. And then the limitation in terms of exposure data is that particularly for non-targeted methods, quantifying that exposure is very challenging. So in terms of the needs from exposure studies to better inform exposure assessment, really ideally we would want quantitative population biomonitoring across all the different chemicals across tens of thousands of individuals, expanding environmental monitoring as well across different media and geography, because if we're doing interventions, it's not just after it's gotten into the body, which was what population biomonitoring is, but ideally we would want to intervene before it even reaches the body. And so that means we need environmental monitoring as well. Additionally, better characterizing the sort of forward and backward relationship between internal and external chemical exposures, including metabolites. And this is still a challenging issue. Most of our high throughput toxicokinetic data is just on the parent compound, and not really looking at metabolites. And even that is limited to less than 1,000 chemicals for which there's data to inform that toxicokinetics. And then to better inform risk management, identifying those subpopulations with greater exposures to chemicals so they can be targeted for intervention, and then ultimately identifying the sources of these environmental contaminants so that they can be stopped at the source rather than waiting for it to get into people's bodies. So with the last few minutes, I wanted to talk about going beyond risk assessment. So there's been an increasing recognition that, really cumulative impacts, the totality of exposures of chemical and non-chemical stressors has the ultimate effect on health and well-being. And this, is almost like a definition of the exposome already as well. And it's challenging to address these sort of non-chemical stressors in the traditional risk assessment paradigm. You could treat it just as another stressor to be added to. You could modify the chemical exposure response, and this is where changing that factor of 10 in terms of the protecting against the variability and susceptibility caused by these other exposures, but that doesn't then get at the direct effects of those other exposures. Or lowering acceptable risk thresholds, saying, well, people who have a lot of populations with a lot of co-exposures, they should be more highly protected against chemical exposures. But this isn't really super satisfying, because first, there's obviously a diminishing return for reducing individual chemical exposures when other stressors are really playing a large role in adverse effects. Just like for lead, if you can reduce lead paint a lot, but if there's more lead in the water, then at some point that you should be turning your attention to the lead in the water rather than the lead paint. There's a lot of data gaps in terms of quantifying exposures and responses that prevents traditional risk assessment from moving forward. Some of these non-chemical stressors are really lack of something positive, like lack of green space or lack of access to healthcare, and doesn't really fit into a risk-based paradigm in terms of exposure to stressors. And then finally, I think, and most importantly, there's dissatisfaction amongst many communities and populations about how risk assessment has or has not really performed in terms of addressing their concerns. Risk assessments can take decades to complete. Individual risk decisions are made in isolation and don't really account for these cumulative impacts over time. And really, a lot of them are tired of being studied, told how many stressors they're exposed to, how many adverse outcomes they experience. They really want to move to solutions that will actually improve their health and well-being in the shorter term. Just an example of the length of time it takes risk assessments to complete. So this, was using Bayesian population approaches for population toxicokinetics, published in 2009. And then while I was at EPA, I led the completion of the toxicological review for trichloroethylene. That was completed in 2011. I left EPA in 2015. In 2020, the Toxics Office finished their risk evaluation of trichloroethylene. In 2023, there's a proposed rule for banning trichloroethylene. There was a final rule at the end, December of 2024. But, of course, the story doesn't end there. Even after the final rule was published, there was legal challenges. There was temporary additional delays in the effective date of the rule. There's administrative stays that was then lifted. And then, most of the rule has been implemented, but the effective date of these specific 6G exceptions have been delayed until November. And so the story still hasn't ended. And it's been, since 2009 when sort of that more novel research was published, and, we're in 2025 and it's still, in terms of implementing actual decision making, it's only just beginning. So this brings me to the National Academy's report that we just released last week on cumulative impacts assessment, and sort of looking at a different approach to try to inform decision making. I really wanted to have a call out to all the committee members listed here, from across the country, including many exposome experts, and then, of course, the National Academy staff that was instrumental in getting this report completed. And I haven't listed them here, but we had a community and tribal liaison group that was engaged throughout the process of the report, informing a number of workshops that we held, as well as a couple of those liaison members served as anonymous reviewers for the peer review of our report. So the major sort of conceptual paradigm change is this sort of cumulative impacts paradigm that we developed. And here, the stressors are separated from resources. We thought it important to distinguish between stressors that cause or promote disease and distress, and then resources that actually promote health and well-being, things like healthcare and social connectedness and socioeconomic factors, things like that. And then the other aspect is that these stressors and resources all occur at many different scales, both spatially from global, like changes in the weather, to the family and individual level. and as well as throughout time, from the past through the future. A lot of the communities we talked about, generations of stressor exposure and lack of resources that has accumulated over generations even, and not just something that is to which they're being exposed at the current moment. And then also expanding the conception of health and well-being beyond just physical health and disease to include mental, emotional health, material well-being, social well-being, and spiritual aspects as well. Now, I'm not going to read this, but we recommended sort of a 5-step process for developing cumulative impact assessments. And throughout this is the preeminent role of meaningful engagement with community, and that ongoing meaningful engagement is informing all the different steps of the cumulative impact assessment process. This concept of stressors and resources is really predominantly drawn from the external exposome. So all these factors that are considered the external exposome are really the factors that most of the communities that we talk to are concerned about in terms of having effects on their health and well-being. And then there's potential for decision making at many different scales, but again, particularly when engaging with a lot of these communities, they're not talking about individual level interventions. They're talking about scales at their community level, supporting their families, supporting tribal traditions, things like that, and then improving the environment. So this is really about external exposome and decision making at the external scale above the level of the individual. So with that, I just want to close by sort of returning to, I think this might be the first of this webinar series where David Bauschaw talked about the exposure science and exposome program. This is just a screenshot. And I really wanted to shift the focus from building tools to discovery to building tools for decision making and decisions. And so in terms of these tools and applications, I think I've tried to argue that characterizing the internal exposome relationship, supporting mechanistic insights organized by key characteristics, as well as in dose response, identifying susceptible populations and quantifying that population variability in toxicokinetics and toxodynamics, sort of making sure that these technologies and tools can be applied in these contexts. In terms of exposure assessment, really expanding quantitative environmental and population biomonitoring, better characterizing that external to internal chemical exposure relationships, identifying highly exposed populations, not just highly susceptible populations, and then what are the sources of these external exposome for which there can be intervention. In terms of, I suppose, moving from risk assessment to cumulative impacts, really it's about characterizing these stressors and resources, both chemical and non-chemical, across spatial scales, across time, and both qualitative and quantitative data being incorporated. And then for health and well-being, again, going beyond disease to the multiple dimensions of health and well-being, and again, having both quantitative and qualitative measures to be investigated. And along the way, always having meaningful engagement. So I'll close with, I feel like the future is now, or at least soon, in terms of being able to apply exposomic tools, technologies, and approaches into environmental health decision-making paradigms, just not at the level of personalized and individual decision-making. I think also in talking to a lot of these disadvantaged communities or overburdened communities, the idea of adding more burden in terms of personalized decision or personalized medicine is just not realistic or even ethical for these communities that already have lack of access to healthcare or already have a lot of other burdens on them, and burdening them with additional responsibility for their own exposome, I just think it is misguided. In terms of the chemical-centric risk assessment paradigm, I think I've already talked through these three aspects of hazard ID, dose response, and exposure assessment that can be better informed by exposome data. And then the engagement-centric cumulative impacts paradigm, really better assessing those stressors and resources at different spatial scales, and then better characterizing health and well-being across multiple dimensions that are of importance to those affected. And with that, I think I'm sorry I'm 1 minute over, but I'm happy to answer any questions.
That's a really fantastic webinar. Thank you, Dr. Chiu. Very educational and inspiring. The floor is open for questions. If you can type questions in the sidebar, I'll read it and we can discuss it. So I have a question. So how do we implement this mitigation strategies in clinical and hospital settings? Like, how do we engage more clinicians? I always feel like that's always the left out thing, right?
I think, a lot of these communities, they don't have a great access to clinicians. So it's really those community health, centers or clinics that they are primarily interfacing with. A lot of them also, even though they are, socioeconomically disadvantaged, they have strong social connection either through faith-based groups or with, social organizations. So I think, having, that interface, I think, is a way to, be in better, contact with sort of that sort of lived experience of those people with these additional cumulative burdens. But I know clinicians themselves are already overburdened with all these different things that they need to worry about. From insurance reimbursements, paperwork. They're being asked to do many different things as well. So I think I just worry that it's adding more things to people's plate is not an effective, necessarily use of their resources, but maybe providing support to them, not asking them to do it, but having someone partner with them might be the best way to go.
Yeah.
I see a hand raised.
Yeah. Can you unmute yourself?
I've allowed their mics.
Okay,
so
does this work?
Yep.
Okay, great, thank you. I was just wondering, this is very exciting. Can you comment on how this can help us address misinformation and correcting risk perception? For instance, something like mercury exposures, knowing that there are multiple different regulatory exposures that can be controlled, but then there are individual personal habits that time activity patterns, things that people do for cultural reasons, for instance, that affect their exposures, and people are not aware of this. So could you comment on that, please?
Yeah, that's a tough one. So in the National Academies report, we talked about, we went through, there are 8 different case studies, some of which are sort of place-based and community-based, where really trying to understand all the different, practices and, concerns in the community first, sort of listening first before trying to, educate or provide additional information. But it's a very tough environment, and I'm afraid I don't have great solutions. But I know and this is not a specific area of expertise of mine, but there's many people who have a lot of experience in community-based participatory research and community engagement. For people that I work with, like there's sort of community advisory boards that are set up with community members, to engage with researchers, and as a way to really bridge that communication. But sort of starting by building the relationship before trying to address a particular issue, I guess is what I found, what I've observed to be to work better.
Thank you.
I don't see any other hand raised. So I guess we can conclude the webinar. Thank you, Dr. Chiu, for this fantastic presentation. And thank you, everybody, for joining.
Yep, my pleasure. Thank you very much.
And thanks.